So let's start our next chapter, that is molecular basis of inheritance. This chapter is fully about DNA, its structure, then its discovery, then the functional aspects of DNA. So the basic things we need to know about this chapter is, we should know what are nucleic acids. There are two types of nucleic acids in the body of various living organisms. They are RNA and DNA. RNA means ribonucleic acid and DNA means deoxyribonucleic acid. Now, what are the basic units of nucleic acid? Nucleic acids are very large molecules. So they are formed of individual smaller units. We call it as nucleotides. നമ്മളൊരു മുത്തുമാല എടുത്തു കഴിഞ്ഞാൽ ആ മൊത്തത്തിൽ നമ്മൾ മുത്തുമാല എന്ന് പറയും. പക്ഷെ ഓരോന്നും മുത്ത് ചേർത്ത് ഉണ്ടാക്കിയിരിക്കുന്നത് എന്ന് പറയും പോലെ the DNA and RNA are long molecules. They are formed of smaller units called as nucleotides. Nucleotides that makes RNA are called as ribonucleotides, and nucleotides that make DNA are called as deoxyribonucleotides. Now, each nucleotide will have three parts or three components. Which are the three components? There will be a sugar molecule, a nitrogen base and a phosphate group. ഇത് മൂന്നും ചേർന്നാണ് ഒരു ന്യൂക്ലിയോടൈഡ് ഉണ്ടാവുന്നത്. Okay, so each nucleotide is formed of a sugar molecule, a nitrogen base and a phosphate group. Now we have to study each of these three things in detail and how they are joined together. അപ്പോൾ ഇതെല്ലാം ജോയിൻ ചെയ്തിരിക്കുന്നത് ഷുഗർ മോളിക്യൂളിലോട്ടാണ്. So first we will see about the sugar. Before that, nitrogen base. There are 5 important nitrogen bases. Which are they? Adenine, guanine, thymine, cytosine and uracil. ഇതില് adenine and guanine are double ring compounds. They are called as purines. So purines are double ring compounds. Only 2 are coming under that. That is adenine and guanine. Now, pyrimidines are thymine, cytosine and uracil. ഇതില് adenine, guanine, thymine and cytosine are present in DNA molecule and their uracil is absent. So DNA is without uracil molecule. Whereas in case of RNA, thymine is replaced by uracil. So RNA molecule will have adenine, guanine, cytosine and uracil. Whereas DNA molecule will have 4 nitrogen bases: adenine, guanine, thymine, cytosine. Okay, so adenine and guanine are double ring compounds called as purines, and thymine, cytosine and uracil are single ring compounds. We call them as pyrimidines. Now coming to the sugar molecule. RNA is having ribose sugar and DNA is having deoxyribose sugar. So we will take deoxyribose sugar first and we will explain about the ribose sugar also. So this is called a ribose sugar because it's having 5 carbon. So the 4 carbons are coming in the ring along with an oxygen. So oxygen atom is there, then the other 4 carbons are here. So they are numbered as C1 or we carbon 1 dash carbon, 2 dash carbon. So this carbon is the 1 dash carbon, this is 2, this is the 3rd one, this is the 4th one. And the 5th carbon is outside the ring and it forms the part of CH2OH group. Okay, അങ്ങനെയാണ് 5 carbons ഇതിൽ വരുന്നത്. അപ്പോൾ 4 carbons are forming the ring along with the oxygen atom and 5th carbon, this carbon is outside the ring. Okay, is that much clear to you? Okay, so this is about. Now coming to the hydrogen and OH groups attached to it. So in case of deoxyribose sugar, this is the structure of deoxyribose sugar. We can see that there is OH group only in the 1 dash carbon, 3 dash carbon and 5 dash carbon. Okay, the other places it is, you know that carbon is having 4 valency, it should be satisfied and all. You will study about that deeper in chemistry classes. So anyway, this carbon is having 4 valency, it should be filled. So this 1 dash carbon is having an OH group and a hydrogen group attached. And 3 dash carbon is also having OH group and hydrogen. Here also 5 dash carbon is also having an OH group. So OH group is present in case of deoxyribose sugar only in 1 dash, 3 dash and 5 dash carbon position. Whereas In case of ribose sugar, this H, here it is replaced by an OH group. So this, the എപ്പോഴും ചോദിക്കുന്ന question ആണ്. What makes RNA highly reactive than DNA? എന്ന് ചോദിച്ചാൽ the main reason എന്ന് പറഞ്ഞാൽ the ribose sugar present in the RNA is having an OH group in the 2' carbon place. And this makes it highly reactive compared to RNA molecule. അപ്പം DNA കേസിൽ ഉള്ള deoxyribose sugarന്റെ ഇവിടെ H ആണ്. RNA-ലാകുമ്പോൾ the 2' carbon is having an OH group also. ഓക്കെ, ഇനി ഇതിൽ ഈ ഷുഗർ മോളിക്യൂൾ DNA-യിലായാലും RNA-യിലായാലും അറേഞ്ച് ചെയ്യുന്നതിന് ഒരു പൊളാരിറ്റി ഉണ്ട്. മീൻസ് ഒരു DNA, ആസ് യു നോ, ഇറ്റ് ഇസ് ഹാവിംഗ് 2 strands. ഒരു സ്റ്റാൻഡിൽ ഫുൾ ഷുഗർ ഇങ്ങനെ അപ്വേർഡ്ലി പ്ലേസ്ഡ് ആണെങ്കിൽ, വൺ ഡയറക്ഷനിലോട്ടാണെങ്കിൽ, മറ്റേ നമ്മൾ വരയ്ക്കുമ്പോൾ എല്ലാം നമ്മൾ ശ്രദ്ധിക്കണം, മറന്നു പോകരുത്. ഇറ്റ് ഷുഡ് ഫേസ് ഡൗൺവേർഡ്സ് ലൈക്ക്, ഇതിന്റെ ഇടയ്ക്ക് കണക്ഷൻ ഒക്കെ വരയ്ക്കണം. സോ ലൈക്ക് ദാറ്റ് ഇറ്റ് വിൽ കം ഡൗൺവേർഡ്സ് ലൈക്ക് ദിസ്. So the 2 strands is having, one is directed upwards, the other one is directed downwards. The sugar molecules are directed downwards. So അതുകൊണ്ടാണ് അതിന് polarity ഉണ്ട് എന്ന് പറയുന്നത്. Now these are the basic things. ഇനി ഇതിൽ എങ്ങനെയാണ് നൈട്രജൻ ബേസും ഫോസ്ഫേറ്റ് ഗ്രൂപ്പും ഏത് ലിങ്കേജ് വഴി അറ്റാച്ച് ചെയ്യുന്നുള്ളത് അടുത്ത ഒരു പാർട്ടിൽ നമുക്ക് കാണാം.